Hi everyone, in this video we will discuss few points regarding HIV infection and its related laboratory investigation. So first of all, you should know few points regarding the structure of HIV virus. So the genetic material in case of HIV virus is a single stranded RNA molecule. This HIV virus contains two copies of this single stranded RNA molecule. So HIV virus is a RNA virus. And this genetic material, that this single stranded RNA molecules having associated reverse transcriptase enzyme. And what is the function of this reverse transcriptase? That reverse transcriptase enzyme is required for the formation of this corresponding complementary DNA from their single stranded RNA. And this genetic material is covered with a core protein, and the major core protein is known as P24. So P24 is the one of the major core protein which is present within the structure of this HIV virus. And the outermost layer, there are different types of glycoprotein. The important glycoproteins are, one is glycoprotein 120 and another one is glycoprotein 41. So glycoprotein 120 is a globular in nature. So what is the function of this glycoprotein 120? This glycoprotein 120 is required for the attachment of this HIV virus with their target molecule, target cell. Target cell means the cells which is having CD4 molecule. So the main cell which is affected in HIV infection is T helper cell. T helper cell is having CD4 molecules on their surface. So this glycoprotein 120 is required for the attachment of this HIV virus with their target cell. And the second glycoprotein is known as glycoprotein 41. So what is the function of this glycoprotein 41? This glycoprotein 41 acts as a transmembrane protein that is required for the transport of this viral genome to the host cell. So these are the major viral protein. All this protein act as a antigen molecule which initiate immune response within the host cell. So the major viral proteins are, one is core protein, that is P24 protein or P24 antigen, and the second viral protein is glycoprotein 120. Glycoprotein 120 is a globular molecule which is present on the surface of this virus and which is required for the attachment of this virus with their host cell. And the second glycoprotein is known as glycoprotein 41. It acts as a transmembrane protein. So these are the major viral antigen which are present in HIV. And these are the different modes of transmission of this HIV infection. One is sexual transmission, the second one blood transfusion, then transmissions through parental route, exposure to the infected blood and blood product, transplacental transmission, and transplantation of the infected tissue. So these are the different modes of transmission of this HIV infection. So coming to the pathogenesis. So once this virus enters into the bloodstream, they comes in contact with their target cell. So the target cell is mainly T helper cell. That is, this T helper cell is having CD4 molecule. So the major target cell is T helper cell. So once they binds with their target cell, that reverse transcriptase enzyme promote the formation of complementary DNA from this viral RNA molecule. And later, this complementary DNA gets integrated with this host cell DNA. And this will initiate immune response within the host cell and antibodies are produced. So that will take some time for the production of this antibody from the target cell. So that time period is known as window period. So actually, by definition, window period is the time interval from the exposure of HIV virus to the production of antibody. That is the window period. So HIV infection predominantly affecting the immune system. That will affect both T cell function and B cell function. So the affected individuals are more prone to develop recurrent infection. They are also prone to develop some specific cancers like Kaposi sarcoma. Coming to the laboratory investigation part, there are some specific tests for HIV infection. So the first one is the antigen detection, that is the detection of P24 antigen. It's a core protein. So the first viral marker which is appeared in patient serum is the P24 antigen. Later, the patient immune system gets activated and the immune system produces antibody. So antibody production takes some time. So the first viral marker which is appeared in patient serum sample is P24 antigen. That is the first viral marker in case of HIV infection. Other one is viral isolation, detection of viral nucleic acid, and antibody detection. So detection of viral nucleic acid, the method is RT-PCR. So window period, this part is important for you. What is window period? So this is the time period from the exposure of HIV infection to the appearance of detectable amount of antibodies in patient serum. So the average window period for the HIV infection is ranging from 3 weeks to 3 months. That's why HIV infected patients remains negative for antibody detection during this window period. So antibody detection test will be positive only after this window period. So the first viral marker which will be appeared in patient serum sample will be P24 antigen. Later the immune system gets activated. The activated immune system produces antibody. So the first antibody will be IgM type. Later the secondary immune response that will produce as IgG type of antibody, and this IgG antibody persists throughout the life of this patient. And regarding the confirmatory test, in case of HIV infection, the confirmatory test is Western blot. So Western blot, it's a type of protein separation, and separated proteins are evaluated after this electrophoresis. So a test is said to be a positive result, then that result should have minimum 3 bands. That is the criteria for a positive Western blot test. There should be at least minimum 3 bands. So that's all regarding HIV infection and its related laboratory investigation. Okay, thank you.